The long-term vision of what this means is that you will have like the med bed. So the idea of like a bed that you put your body in and it scans you, just like looks at what's wrong with you and then beams you with energy and fixes you, right? It's like very sci-fi, but I think that the science is there.
Welcome to Legible Bio. Thanks so much for being here today, RJ. The point of this podcast is to make biotech a bit more legible to smart folks outside of the Cambridge and South SF biotech bubbles, and most importantly, for me to better understand biotech. The complexity of biotech is as interesting as it is challenging, and so I'm bringing on folks who can explain things in simple ways. And I think you've done a really great job of it in the past from the stuff I've watched and the conversations we've had. And so I thought you'd be a great guest to have on. To start off, why don't we just kind of do a brief background on who you are, what you're building, how you got here?
Yeah, my name's RJ. I have a Twitter named Anabology. I think I'm sufficiently doxed now. It was originally an anonymous Twitter that I grew to a decent size. But yeah, like where I came from, I mean, I grew up in Texas originally, and then I went up to upstate New York after doing like a kind of biochemistry undergrad in Texas because I got really interested in the aging space where back in college, I really didn't know what I wanted to do, but I really enjoyed self-experimentation, like taking nootropics or just exploring consciousness in different ways.
Does that mean you just did a lot of shrooms? Is that what that means?
So I had a class in college where it was this like graduate level class that I was taking as an undergrad, and it was pretty small. So the professor went around to like all the undergraduates in the class and wanted us to like introduce ourselves, and like what they were like, why are you taking this class? And it was a neuroscience class. And I said, well, I'm just really interested in neuropsychopharmacology. And then the professor just like laughed at me, and he's like, yeah, when I was in college, I was interested in neuropsychopharmacology too. So yeah. So no further comments. But yeah, I don't know. I was just really interested in this, like self-experimentation, nootropics even, and I wanted to find all this kind of like information about how to do these things, how to take supplements or make myself smarter, or like explore the different states that I could feel without hurting myself. So I like looked online for all the best information that I could, and I found this forum called Longevity. So Longevity was one of the OG kind of anti-aging longevity forums, but it also had all of this great like biohacker information and links to papers about like these novel compounds that were coming out. And I started digging in there just like from a self-experimentation perspective. And then I realized that these people were talking about like basically being an immortalist, like living forever. And that was when I realized I was like, I have no idea what I want to do, but I'm not having a terrible time right now. So, I mean, I would like to have longer to figure out what I want to do. And I think longevity is probably the right way to go about that. So yeah, that was college. And then I went up to upstate New York to this company called Ichor Life Sciences. It's kind of like an anti-aging biotech and like contract research organization. So what they were doing was like both studies for companies like just big pharma companies or other biotech companies that needed stuff done in the space, and also I was able to do my PhD with them. So they're partnered with a local university, and I was like doing my PhD up there with them. And after a few years, I left that and kind of started my main biotech company right now, just because I had an opportunity to get some funding and like pursue the idea that I wanted to pursue faster. Yeah.
There's a lot to dig into, but I think one of the most interesting things about you is that you've kind of explored the fringes of what is mainstream science. People kind of poo-poo the Reddit forums and all of these other things, but they're increasingly becoming more and more mainstream. But how did you think it was important enough to go experiment on yourself and explore these fringe theories that we're going to get into, Gilbert Ling and all of that? But why did you think that was important, and how did you even start?
Well, so I think the initial like way that I got into it wasn't really out of like some specific affinity towards the edges or like something that's not the mainstream. It was just that the mainstream solutions didn't work for me, where I was like kind of in chronic pain as a child and like super depressed and just like not really having a good time until I started to like do these biohacking things, and like the stuff that doctors would tell me was just completely like not useful. I was having like migraines every single day. I would go to school and like throw up out of pain, and the doctors like put a probe in my stomach with like a camera, and they saw stomach inflammation. So then what they told me was that, oh, it's from eye strain because your eyes are like a little bit bad, and maybe you should go get your glasses renewed. And that was the solution that they told me after seeing stomach inflammation. And I'm like, this is just, it was like obviously not helpful. I mean, that was when I was like a child, so I didn't really have the knowledge to push back against that. But that was, they really just like did not help me at all. And I really only ever made progress on like migraines and like my mood and affect and things like that after doing the things that like ended up being on the fringes. It was like more of a proof of work type thing.
What helped with the stomach pain and the migraines?
Yeah, I mean, I think one of the big things was like seed oils for me, where like just generally, the more seed oils that I eat, the more migraines I get. And when I like eliminated them fully, my migraine frequency like drastically decreased over the next like year of elimination. And I think that is also probably related to like the stomach inflammation. Like maybe my stomach was just reacting badly to that. So I mean, like to me, the main thing that helped was just changing my entire diet. And I feel like that should have been the solution when you see someone's stomach is inflamed, and then maybe it's like what you're putting in your stomach that's hurting you, right? So
Yeah, my med school friends hear me say it, but I just do not trust the medical establishment enough to
mhm.
where I can't trust them to, you know, recommend proactive measures. It's because they always rush to these super intrusive methods of treating you that may or may not work and do
Right.
not are sustainable.
Yeah, and I think the average like a cost in the U.S. that some the average person spent on healthcare in the last year was like $12,000, and all of this is on like the kind of acute treatments. Really, there's like basically no preventative medicines. Like I think Eli Lilly maybe now has like a preventative medicine like the Ozempic or like I guess what I forget which one is theirs. Like
Uh,
they have not Ozempic,
Wegovy
Mounjaro,
and
Wegovy. Okay,
yeah, Wegovy and
yeah.
tirzepatides, yes.
Yeah. And I mean, ideally, like all of biotech and all of health and like the medical establishment should all be preventative medicine because it's like much harder to fix somebody once they have diabetes than to just make them like not eat a toxic diet in the first place and not get to the point where they have diabetes, it seems.
Yeah.
And it's also like much more like monetarily lucrative. So I feel like we're going to get there just from like market forces. Like Eli Lilly just got to be the first like $1 trillion biotech, I think.
Yeah. How did you, or do you want to talk a bit about what you're building now? Speaking of non-intrusive methods of treatment, what is it that you're building now, and how did you decide that's what you wanted to work on?
Sure. Yeah. So yeah, the main thing I'm working on these days is Aion Biosciences. So I'm like co-founders with my friend Benjamin, and what we're doing is we are trying to essentially read and write to the bioelectricity of the body. So the long-term vision of what this means is that you will have like the med bed. So the idea of like a bed that you put your body in, and it scans you, just like looks at what's wrong with you, and then beams you with energy and fixes you. Right? It's like very sci-fi. But I think that the science is there now to tell us like at least how to get there. Whereas like before, if we went maybe 20 or 30 years back, like the science wouldn't have really told us how. Um, so we're pretty convicted that bioelectricity is the path there. So what that means is the kind of like voltage of the cells is like what we refer to when we say bioelectricity, and the way that the cells communicate by exchanging this like voltage information, ions through gap junctions or all of these like direct ways that cells can communicate electrically. A lot of this stems from like the work of Michael Levin, and I'm sure we're going to talk a bunch about Michael Levin today. The kind of simple idea is that every cell in your body kind of works like neurons, but just on a different time scale. And he was talking to Lex Friedman, I think just the other day, about how there's kind of like a spectrum of cells. So when you ask a biologist, like, what is a neuron? They would answer, oh, it's what looks like a neuron. It's like electrically excitable. It'll fire, have action potentials. It looks like a starfish kind of thing. But if you like look at development, there's not really like a perfectly clear boundary when something becomes like a neuron or not, right? They all come from like the same initial like embryonic cells, and then they all differentiate, and then there's like slight differentiation events that occur, and then the barrier between what's a neuron and what's not is not like there's not like a on or off switch essentially.
Yeah.
So then if you take some attributes of neurons and you're like, oh, it changes its voltage over time to process information, well, you can look at a skin cell and it does that, but over hours instead of like the milliseconds. So if you kind of treat the whole body like your nervous system, and instead of just doing this like say millisecond processing of information to react to move your muscles, for example, in your environment, maybe it's doing like an hour-long processing of information in your skin to close a wound, or like a 24-hour-long processing of information to change the metabolic rate in your liver or something, right? So yeah, like this is the framework that we're like looking at. It's something that is this like emergent property of the collective, where if you look at a single neuron, it processes no information. You can't like be like basically on or off. But if you look at a collective of neurons, it can do these like wonderfully complex things, like move the body around in the world, communicate, have social life. So then the rest of your cells, if you want to have these like complex outcomes, like make the whole body feel younger or be younger or regenerate organs, a single cell doesn't really know what an organ looks like or how to regenerate an organ, but a collective of cell does. So that's like the layer that we're trying to intervene on. And the biophysical mechanism of this is voltage, and this is what Michael Levin has worked out.
Yeah. There's a lot to unpack there as well. And I think we're going to talk even more about Michael Levin later on. But in trying to kind of paint a fuller picture of the scientific foundations for what you're building, I realized there's, I think, 3 key players to understand in the space. First is Gilbert Ling, second is Raymond Damidian, and third is Michael Levin. And so I kind of want to go through each one of them and their theories and their works, and then kind of figure out how all of them fit together, because some of the stuff I read about Gilbert Ling does not necessarily square with everything I understand about Michael Levin's theories. Let's start with Gilbert Ling. He's somebody I found out because of you. He's been influential in your worldview. My understanding of Gilbert Ling's work is that he kind of tried to disprove the fact that cells have membrane pumps, and suggested something else entirely. There's a couple quotes from you that I want to read out just to set the context here. First is, if you're a biologist, you're almost always a taxonomist. You get into biology because in most cases you simply didn't like math or chemistry, but you wanted to be in the sciences in some way. You do biology because you have a good memory and you can memorize the taxonomy, and that's required, which is the status quo. This has been the culture of biology for a while. And a related second quote is, anyone can imagine what a pump does, but no one can imagine electron density. What you're trying to get at here is that we impose these structures of proteins that are human legible onto biological systems because that's been the culture of biology. The people who do biology aren't mathematicians, physicists, whatever. So it's harder to imagine alternative models. And Gilbert Ling is proposing an alternative model for understanding how ions move between cells. Is that the right way to understand it, the right way to look at it?
Yeah, I think that's like pretty accurate. I really think that one of the reasons why Gilbert Ling's work didn't take off was because of the difficulty of the work. It's like just a pain to get through. It's like not an easy book to read.
Yeah.
If you read any of his books.
I tried. I ended up reading it. Like it was hard. Yeah.
Yeah. So, yeah, I mean, maybe to give context, I can just go into like who Gilbert Ling was, like what he did and what he said, and then we can get back to like the taxonomy kind of argument, and maybe like why Gilbert Ling's things like didn't really pan out into the mainstream. Yeah. So Gilbert Ling, he was this Chinese biologist who came here from China in the, I think, early 1950s, on this Boxer Indemnity Scholarship. So it was a scholarship where China basically chose one person out of each category, like physics, math, chemistry, biology, and gave a scholarship to one student who tested very, very well in these categories, and then sent them to the US to do graduate studies. So he was the one who was selected for biology for this. And then around the same time, the physics person who was selected for this was this man named C.N. Yang, who eventually won the Nobel Prize in Physics for making Yang-Mills gauge theory, essentially, which is what some to consider to be the last big revolution in physics, which kind of unified the electromagnetic and strong and weak like nuclear forces, or I guess electromagnetic and weak nuclear forces, and added the strong force to this description. So then Gilbert Ling was like college roommates with C.N. Yang, and I think got some inspiration from him and like worked on this like more physics-based theory of biology alongside him, and instead of just this kind of like taxonomy-based theory where you're just saying that there's like a pump or there's like a hole, then things go through the hole and that's like all we describe. He actually talked about everything mostly from this, it was like a more like kind of quantum chemistry perspective, where you're talking about like the electron density of the proteins, or like the way that like molecules can withdraw or donate electrons to proteins. And the reason why he did this is like during his grad school, he was tasked to do this graduate seminar when he was like working in this very nascent field of bioelectricity, essentially, like the idea that cells have voltage gradients across the membrane, or they have ion gradients across the membrane. And he was tasked to research and give a seminar to his graduate school on the sodium pump, which was the common idea that hadn't been proven yet, that there was a pump in the membrane that essentially actively pumped the sodium out of the cell to keep the sodium like at a low concentration in the cell to maintain the voltage of the cell. And he did all of his research like in the libraries at that time and like looked at all the data, and he's like, wow, there's like actually no evidence for this thing at all all of this data is like totally fake. I mean, nobody had made a crystal structure or anything yet, so he was looking at an earlier version of the evidence, but at the time he was very convinced that it was not real and not consistent with the evidence. So then he went to the seminar and basically said that. He's like, I have nothing to report. This is, there's nothing here. And afterwards, his professor or his PI, right, the mentor of his PhD, pulled him aside and was like leave this one alone. Don't make yourself a martyr because this sodium pump is a sacred cow. People like have staked their careers on this. And then he basically said, fuck you. I didn't come to, America to worship a sacred cow. I came here to do science. And then he kind of spent the rest of his career, maybe out of anger or something, but basically fighting against this idea of the sodium pump, for better or worse. I mean, there is some data now that, he does later in the career acknowledge that this protein does exist and has specific attributes. But during the time that he was kind of denying the sodium pump, he did make like these really great advancements and made like a totally alternative theory of life that I think is maybe even more consistent than the mainstream theories that we have now. And I can go into what that theory says if you would like.
Yeah, he has a really interesting life, but the theory, my understanding, the simplest way for like a real layman to understand it is that he said a cell is not a sack of salty water. It's more like a piece of jello. And that, intuitively makes sense, but it has all sorts of implications. So what are these implications for looking at cells in such a different manner?
Right. Yeah. So the fundamental thing is what he's saying is that the proteins in the cell and maybe even on the cell surface are more important than the membrane itself. So like the idea is, mainstream idea is that the cell's like a water balloon where if you want to have different amounts of salt on either side, you have to poke holes in the water balloon for like the salts to diffuse through. And then because there's constant diffusion, you have to have like a pump to fix whatever went wrong with your diffusion. But then what he says is, like you said, it's more like jello, where instead of it being like a water balloon, like imagine you have some like uncolored jello in solution, and then you put this in water, and then you put like a dye into the water, but the dye really likes to stick to the jello. Well, then the jello is going to accumulate all of the color, and your jello is going to be a different color. That's kind of the idea, but with ions. He's saying that the proteins like inside and on the cell can bind to the charge carriers, the ions, and then create the voltage gradient as like an equilibrium process rather than like a non-equilibrium process, if that makes sense. So it's like not consuming energy to do so, but it can consume energy to create like state changes. So the implications of this, it basically just gives like more mechanistic insight into like what biology is doing. So if I like look at a cell, right, and I say the voltage is this, well, all that tells me is that there's some like difference in ions inside and outside the cell, and there's like no clear connection to how the machinery of life should behave at all, right? Where, well, if the proteins aren't binding to the ions and there's like not significant interactions between ions and the proteins, then how is it affecting how the proteins are functioning in the cell, and how is it affecting the way that like life is behaving? But Gilbert Ling says that all of life's process is basically the interaction between proteins and ions. So one of the analogies that he used is you can take a red blood cell, and red blood cells are basically just these sacks of hemoglobin. They're like basically mostly hemoglobin in weight with water as well, like associated with the hemoglobin, and some salt, and that's about it. They've like lost their nucleus, they've lost like most of their machinery. So then you can kind of cut a red blood cell in two and then rejoin the two sides like with itself, right? And then you have two smaller red blood cells. And then you can cut those, and then you get 4 red blood cells essentially. And then you can keep cutting these down until you get what he called the nanoprotoplasmic unit, which is a single unit of hemoglobin, the water around it, the ions that it has bound to, and then whatever other molecule can be like the trigger of life, right? Like say you have ATP that can alter the conformation of the hemoglobin. And then he's like, this is the fundamental unit of life. The cell is not the fundamental unit of life, but like the protein water complex is like the base unit of life. And then if you like look at it from that perspective, then you know exactly what happens whenever there are voltage changes in the cell, right? There's a different, essentially, he calls it C value. One of the reasons why Gilbert Ling's work is so hard is that he also introduces like a ton of like new terms that are not standard. Essentially, the more mainstream version of this is conformation. So if you take an ion and you bind it to these sites on a protein, like the negatively charged carboxyl sites on the side chain of the protein to be precise, but if you bind potassium to a protein, the shape of the protein will be different than if you bind sodium to the protein, because it interacts with the like electrons that are being pulled towards the positive ion slightly differently. So then the transition that he sees and what he calls life's action is that there is this fully kind of unfolded protein that binds to potassium. And then this also associates with water. So then you have this like water protein complex that alters essentially the solubility of the things around it. So it makes like a force field in water where if I have like all of this like structured water on the surface of a protein, and then I try to put, another molecule into this like milieu of structured water, the water is going to exclude the solute. So it kind of creates a force field around the protein that like keeps it isolated from the environment a little bit. And then whenever you remove the potassium, the protein likes to fold in on itself and bind to other charge groups on its, the protein itself, and also likes to bind sodium more. So then it collapses into a folded state and then releases the water. And whenever you release the water, this is like a big, essentially like entropic gain. Like you increase entropy by a lot because the initial state was like a low entropy, like high order state. And then this active state where you folded the protein in on itself and lost all the water is a high entropy state. So this is a very favorable, like thermodynamically transition to happen. And he says that life can use this specific transition to like do all of these like really complex things. So then, this is like a kind of complicated thing, and it's like very like mechanistic and in the weeds, and it doesn't seem like on the surface that profound. But then you can take this and then reason through like what should happen if I say change the salt concentration inside a cell, because the structure of the water changes the volume of the cell. So if you see cell swelling, then it can be correlated with voltage, for example. If you see, say, some like transcriptional change, right? This may be from like the chromatin remodeling in the nucleus. So in the nucleus, different genes will be exposed so that different machinery can be made, so the cell can do different things. But this is dependent on the folding state of chromatin, which is also dependent on the salt concentrations and the ion strength and the voltage of the cell now under this framework. Whereas chromatin, it's essentially these like proteins that can kind of structure the DNA, right? So if you have like chromatin that's like all opened up, this is most likely more like a stem cell where a lot of things are being made, and then if you like kind of like fold the DNA all nicely, and you have like really specific segments of DNA exposed, then you may be able to have like some very specialized cell that only needs these specific proteins. So then this is like a mechanistic connection between voltage and then cell fate. So like what the identity of cells are. So back to the question of like, what is a neuron? Well, a neuron has this like resting membrane voltage of like negative 70 millivolts. And then a liver cell or something else, right, may have a resting membrane potential maybe around like negative 50 or 40 or something, right? And I'm getting these numbers a little bit wrong. But the question is like, why does that happen? Like why would just changing the voltage of the cell change the identity of the cell? And then I guess I'm saying here that Gilbert Ling kind of has an answer where you're saying that I'm changing this global parameter, which is how tightly bundled up these proteins are, like in both the cytoplasm, the membrane, and the chromatin. And then this directly changes the transcriptional state of the cell, which directly changes the cell identity. It's like so Michael Levin is this top-down thinker where he's thinking about, how do we apply the tools of like sociology and psychology, but to more inanimate systems, like or animate systems, but like simpler like cellular systems or even subcellular systems. So he's coming top down. Gilbert Ling is like bottom up. It's like how do you build from like the most fundamental possible unit of life and then get this like emergent complexity.
So what I'm hearing is that Gilbert Ling is not actually challenging the central dogma, which is how proteins are expressed, how DNA is expressed rather from DNA to RNA to proteins. He's proposing an alternative theory of how this central dogma might actually work.
Yeah, right. So yeah, he didn't challenge the central dogma, and like Gilbert Ling overall, and this is one of the things that probably made me trust him is generally like a somewhat of a science like conservative in a way, not like conservative politically, but he was, he's a big proponent of like, say, he thought climate change was going to kill us, as like, and he was pretty vocal about that, and he supports like the importance of DNA and central dogma and everything. But he like the one thing that he just could not get over was the origin of the voltage gradient across the cell. He thought it was specifically like this equilibrium process of proteins binding ions and not this active pump in the membrane. And there's some data to support that. I think the most interesting one that I'll mention is he was able to take these muscle cells and then cut them in half and then look at the open half on electron microscopy to see that there was no membrane covering the open half. It was pure cytoplasm. And then, so basically, you have a cell that's like open like this. And then the conventional model is that you have a barrier around the cell and ions diffuse in and out, and they're only diffusing in and out dependent on basically the amount of holes that you poke in there. So then if you opened it up, it should totally just depolarize the cell. All the ions should float out, and then the membrane potential should go to zero. But then he did that. He had a completely open cell that he made stable, and it didn't go to zero. It was still like a significantly negative membrane potential. It lost some, right? It lost maybe half of its membrane potential, but not all of it. So I think that the ending kind of model will be eventually a merging between Gilbert Ling's model and then the membrane pump theory. Like I think that the pump is better explained under a Gilbert Ling mechanism where there's like dynamic binding and unbinding of ions to the pump dependent on the electron density of the pump. But there are also these like crazy feats of data that just can't be denied, that like you can cut open a cell and it doesn't lose its voltage. And it's like, how does that work?
Is that similar to the experiment you actually did where you replicated some of Gilbert Ling's studies, the super simple
Right.
experiment?
Yeah, it is pretty similar. So what we did is there was like a super simple experiment that he proposed that even high schoolers could do.
Yeah.
Basically, you just take a dialysis sac, which is like a sac that has really big holes in it. Basically, everything can diffuse through it except, say, really big proteins. So you take proteins, you put them in this like dialysis sac that's like totally permeable to ions. It should be 1-to-1 membrane potential across the barrier. And then what you do is you measure the salt concentration on inside and outside. And then the experiment is to acidify the solution. So you just add basically protons into the solution. And then under Gilbert Ling's model, these protons should bind to the hemoglobin, withdraw electrons into closer to the protons, and then change the state of the hemoglobin, which should now associate with more water and exclude sodium, was the idea. Because now it's like associating with water and it's having the force field around it that I talked about, right? There's this like force field that excludes ions around the protein now, just made out of structured water. So then we did that. We took the hemoglobin, put it in a sac, acidified it, and then we found that this simple solution of proteins excluded sodium without a pump. And the mainstream model is that you need a pump to push out the sodium. So yeah, just it took like less than $100, just super simple to do, and it seemed to work.
And this is because red blood cells don't actually have membrane pumps.
Oh no, so this is like a model of a red blood cell. Like I think red blood cells like do have the pumps in them still. Gilbert Ling has done a bunch of experiments where he's tried to you can make these like red blood cell ghosts. I think they're called like white ghosts and red ghosts. Basically, you can like destroy a red blood cell, and then you can isolate just the membrane and then reform the membrane into a cell-like object. And then you can also destroy a red blood cell and then get rid of the membrane and only reform the proteins into like a cell-like object. And then he like measured the voltage of either of these, and he found that the ones with the membrane only couldn't partition the ions, but the ones with the proteins could. The ghosts. So that's like another direct thing for red blood cells. But this one is like an even simpler model where you just take the hemoglobin, not even like the other components of the red blood cell without pumps. There's like never a pump involved, and then you just put it together in a sac that you made and find these results. And I mean there are possible like other explanations of it, but there's like so much data. I think it's like worth understanding if you're on this like mechanistic side of biology. If you're trying to understand like the deep biophysics of biology, I would recommend reading Ling's work, because I don't think anybody's gone that deep with at least the level of data that they had at that point.
I promise I tried to read it and understand it. It takes time. It takes time to kind of wrap your head around it. I guess the bigger question is what it means for medicine in the future if his theories are correct. I think now is a good time to talk a bit about Raymond Damidian, who I believe was inspired by Gilbert Ling or worked with him in some capacity. Raymond Damidian is the uncredited inventor of the MRI. Is that right? And a big influence on how you're gonna build your own company. So why don't you tell us a bit more about Raymond Damidian and why he's important?
Yeah. So yeah, Raymond Damidian, he was another person that was kind of generally interested in bioelectricity back in the 60s and early 70s, where he's originally studying like the kidney in St. Louis, Missouri, where I am now, because it was the organ of bioelectricity, right? Like it regulated the ions, which create the voltage gradients in the cell. And eventually he kind of got a similar quest as Gilbert Ling, where he was supposed to isolate the sodium pump. But he like couldn't do it. They were having a lot of trouble with it. So he started like searching out these other theories of life and how like voltage could be generated. And he came across Gilbert Ling. And what Gilbert Ling said, of course, is that the voltage is going to be correlated to the structure of water inside the cell, right? So, like I said, the state of the protein changes the voltage of the cell because it changes the way that ions bind to the proteins. And that change, and then the protein interacts with water in different ways depending on the voltage. So then when he saw this, he realized that he could measure it, where you could look at essentially the state of water in cells, like non-invasively, with this technique called NMR. It's like nuclear magnetic resonance, and you're looking at these spin properties of protons in the water. So you can directly probe the water with this technique. So then he took cells and he started like looking at different ones under NMR. And specifically, he took healthy cells and cancer cells because he knew already that the voltage was different in these healthy and cancer cells. And then he was able to detect that there was also a water structure difference between the healthy and cancer cells. And then that was like the big light bulb moment where he realized that because this is true, now you could make a machine where if you just did this technique that is basically zero dimensional in 3D, then you could like image the body like non-invasively and detect cancer. So then he published this in 1971, like the results that cancer cells and healthy cells have different water structure. And he proposed the idea of the MRI then. And then over the next like few years, he basically did the first scan on animals. He did the first scan on a human. He did it on his like grad student because he was a little bit too fat to fit into the scanner by the time they made it. Yeah, he did the first like MRI scans essentially. And then, they kind of rebranded from over time from NMR, which is like nuclear magnetic resonance, to just MRI, which is magnetic resonance imaging. They dropped the nuclear because nuclear was kind of a swear word at the time.
Yeah, sounds way
So
scarier.
yeah. So then, he made this machine, he made the MRI, and it was like a big revolution. Initially it was like pretty like low resolution and slow, where it didn't have a ton of clinical utility, and a lot of people criticized him for many years. But he did make one of like the most important machines of all time, basically from one of the insights of Gilbert Ling's work. And then eventually you say he was like uncredited. So what happened is so the first thing that happened is like after he made the MRI, he had the patent for it, and then there were within a year after he sold the first one to a hospital, there were more MRIs from companies like General Electric than from his own company, because they just decided to violate his patent and try and win in court and get the profits, before litigation. Yeah, so that happened, and he kind of just like spent the rest of his career like litigating because many people did this, and he was like a small company and kind of academic background rather than like this, corporate, cutthroat background. So that was like one of the first things that happened. And then later he started to become like more and more vocal as like a Christian and creationist. Like he believed that the world was created in 6 days and denied evolution, and was very vocal about that. So then he kind of undoubtedly created the MRI, and he was recognized by even presidents that he did this before. But then eventually like the Nobel Prize was given to somebody else for the MRI's creation. It was given to Lauterbur. And Lauterbur was this guy who basically what he did was he used magnetic field gradients to encode the MRI signal. So he just did some innovation that they made the MRI much faster. Whereas like he varied the magnetic field, whereas to get the spatial localization, whereas Damidian just varied the position of the person in the MRI to get the spatial information. So Lauterbur made the MRI much faster, but he didn't invent it in the first place. So the Nobel Prize was given to him, and it seems kind of like wrong to me that it's not given to the inventor, to somebody who just like made some kind of engineering innovation that made it more clinically viable. But you could argue that maybe this guy still should have it because it was a big innovation. But the Nobel Prize can go to 3 people, right? It can go, and then the prize was only given to 2. It was given to Lauterbur and then a colleague of his. So the specific omission of the third person of Damidian was like a big FU from the Nobel Committee to Damidian specifically, because he was a vocal Christian, is one of the most likely stories in creationists, and specifically denied evolution. And the thing that supports this is that in Lauterbur's Nobel-winning speech, after he received it, like his acceptance speech, he, after being an inorganic chemist for his entire career, not a biologist, said that the next thing that he was going to work on was evolution. So yeah, so it really seems like he wasn't given it because of his evolution stance. And then also there were issues about him like trying to lobby for it, right? He was like saying that, I deserve the MRI or the Nobel Prize for the MRI, and then maybe they really didn't like that. So then his career kind of was like derailed because he spent like most of it litigating instead of like developing, more things. And then he held his hat on this kind of non-mainstream theory of biology that other people didn't really support, where eventually he did. So Gilbert Ling lost his funding later in his career because the government agencies were no longer interested in it. Some of his enemies made their way into the funding agencies, who, supporters of the pump theory. And then Raymond Damidian, having at least a little bit of a fortune from his company, funded Gilbert Ling's research for the rest of his career. So he was like writing on the back of this like non-mainstream theory. He was litigating all the time, and he was just, I guess, crying about the Nobel Prize to a degree because, yeah, he like lobbied for it pretty significantly and was denied it unfairly. So his career was like pretty derailed, and the long-term vision that he said that he wanted for the MRI was not just a machine that read, but a machine that essentially read and write, like a cancer-killing machine is the vision that he had for it. But it seems that, he only said this, he never actually executed on it. And then, I had read Gilbert Ling and I saw Raymond Damidian's story, and I saw that this was like an fulfilled vision, that there was like no real fundamental barrier in front of it happening. So I thought that I should just do it. And that's what prompted me to start my company.
That's fascinating. I didn't realize the connections between Gilbert Ling and Raymond Damidian were so deep. How does all of this connect to Michael Levin's work around bioelectricity, and how different is the Ling view versus the Michael Levin view?
Right. Yeah. So I think that Gilbert Ling, you could have gotten here like a lot earlier if people like believed in Gilbert Ling's model. But I think Michael Levin has translated this idea that bioelectricity can do these like really crazy things into this like mainstream view now. So yeah. I mean, Gilbert Ling, like an extension,
bioelectricity legible, like that.
That's exactly.
Kind of what he did. Yeah.
Gilbert Ling was pretty illegible, but if you like really went down the theory, you would have seen that it, altered cell fate. Like I think Gilbert Ling did describe development as a sort of gradual excitation of the cell. Like the voltage changes across, cell development, for example, like turning a stem cell into a skin cell into a bone cell. So, but Michael Levin really popularized this. So some of the crazy things that Michael Levin has done, he's taken some melanocytes. So it's like the cells that you find in moles, like the brown cells on your body. He's taken one of those, and then he just changed the voltage of it to be the same of a melanoma. And then the cell turned basically like into a melanoma-like cell, where it started to form tumors and migrate and do all the things that a cancer cell would. And then he turned the voltage back to that of like the native resting melanocyte, and then it reverted the cancer-like phenotype. So he's like created cancer and gotten rid of it. He's done these crazy things where he's made eyes appear in different parts of the organism just with like bioelectric changes. He's regrown limbs. He's made different morphologies where like heads that have one head worms that have one head genetically, he made have two heads with no genetic changes.
Yeah, that stuff is just so crazy. How is he actually manipulating the voltage? Is he like putting a wire into the petri dish? Like, what is he doing?
No. Yeah, so there's a few ways you can do it. It depends on like how much spatiotemporal control you want of how you do it. One way you can do it is you can just change like the salt concentration in the solution around the cell, and that should change the voltage. But then if you do that in the body, you can't just like really do that because the body maintains its like salt concentrations very well. So another way you can do it is you can do like RNA injections like into cells, where you can say code for like an ion channel to be expressed here, or you can genetically modify cells with some like say knockout ion channel, where I can make a circuit that expresses an ion channel that changes the voltage of my cell, and then I can remove the gene when I want it to go away, or things like that, right? So it's like mostly genetic tools. The chemical tools like salts or drugs that can close and open ion channels like are used, but those are more like global nudges because you can't really like localize them to one very specific place easily. So yeah, for like spatial precision, genetic tools are used, and then for ease, the chemical tools are used.
And what you're trying to do is have spatio-temporal control over how the ion gradient in these cells is manipulated, and you're using the Damidian MRI approach of being able to write cell states using fields. How does that even work? How is that feasible?
Yeah. So, well, I guess the first thing I want to say is like that if we achieve this, the reason why we are convinced that it will be useful is because Michael Levin has shown all of these crazy things where you can basically change the fate of cells and the identity of cells just by changing the voltage, right? Yeah. So it doesn't really matter if it's like a chemical or a gene or like a field externally. All that matters is that you're changing this like global kind of volume knob of voltage, and then you get these crazy like cell and like organism level outcomes.
Mm-hmm.
And that's like kind of one of the core reasons why we think it's possible now, is that previously there has been this research on how do we interact with ions in cells using external fields. But the question is like how does that become useful if I'm doing this in the 90s and I don't know the consequences of bioelectricity, right? So what we're doing is we're taking like this kind of previous work that has been done where people have like looked at ways to alter ion concentrations in cells with fields, and then we're pairing that with like the current knowledge that has really been made legible by Michael Levin, and then saying, like that if we apply these in a bioelectric context, we'll get these outcomes. Like it's a fairly simple thing in the end, but the really hard part is like that this field is like not replicated well. So one big side of the field has been centered around this theory called ion cyclotron resonance. And it's this idea that very low frequency and low strength magnetic fields can alter ion flows when they're tuned to a resonant frequency of the ion. And the mechanism kind of like makes no sense. I could dive into it, but what I'll say about it is that it's kind of like a battleground, or I don't know if a battleground, more like a graveyard. Like even without a battle, like people like make these like physical theories of biology where you will say that because I'm getting this effect at this very low field strength where I'm applying a Earth's level magnetic field to cells, and I'm getting this crazy outcome where I'm seeing the calcium flow out of cells, like some ion flow out of cells. The only way that can really happen is from like a quantum mechanism, because energies involved with these magnetic fields are below the thermal noise floor. So there's like imagine I'm trying to blow on a leaf in a hurricane to make the leaf like go into my house or something, right? That's like the level of energies that are involved, right? Like the thermal noise inside of a cell is like it's like a hurricane, and then the ions are like leaves being like tumbled around in the hurricane, and then the magnetic field is like a small like breath that you're just like blowing onto the leaf. It's like doesn't seem possible. So then the only way that you can do it is you can't work on this like classical level where you're physically moving the ions. You have to change something about like the essentially like phase of the ion. You have to change a quantum property of the ion to alter a reaction yield, for example, or like some tunneling pathway, like things that rely on much lower energies. And then the big problem is that biology is like warm, wet, and noisy, right? Like the thermal noise, even though you can kind of get away from that by using like a quantum mechanism, like the energies align better. The problem is that the hurricane is so strong that it decoheres the quantum state like extremely fast. Like immediately the quantum object, because like everything is a quantum object, interacts with its environment and loses its quantum nature. So then, the big question is like, how do you actually get a situation in which the coherence time, like the length of time an object can behave as like a quantum object, like how do you have that be long enough to be relevant to anything in biology? And then that's kind of like what all of the theories have centralized around in like magnetobiology specifically. And people always like end up finding issues in it. It's like there's maybe less than 20 people in the world like actively developing these theories, probably even less now. I think most people are dead who were working on these. And people will like be like, oh, I've solved it. And they go to a conference and be like, I've solved magnetobiology. I know everything. And the next year they'd be like, oh, I was totally wrong. I found it. I found that I was off by a factor of 2 at least. So then it's just like it's so unsettled and barely anybody's working on it. Like I think that there's like a stigma around quantum biology in general, but it's the only possible way that you can get some of these like at least magnetobiological effects. So I want to like zoom out for a second. When I'm saying using fields to write to cells, like one big way is you can directly interact with the ions through these like quantum effects with magnetobiology, right? So there's a few theories around it, like ion cyclotron resonance, ion parametric resonance, etc. These things that could be dove into, but then it's unknown if these things will replicate or can replicate. I mean, people have replicated these before, but then who knows if there's a replicating artifact. So we're being a little bit agnostic to the exact how. And the dream of read-write MRI is like the most clean with magnetic fields because it is like a big magnet essentially. But we're also looking at ultrasound, and we're looking at like other possible ways that we can solve this problem. So like a different version of it could be that you take, say, a robot arm that has like some focused ultrasound array or ultrasound array on it that can read in high fidelity whatever parts of your body, and it'll just like go over your body and like scan you, and then notice when something's wrong, and then use the ultrasound that's already built onto the robot arm to fix whatever's wrong, right? So there's like many versions of this, but the core idea is reading and writing to bioelectricity. And then And then the reading bioelectricity part is the part that people get hung up on as well. And this is where the history of Gilbert Ling and Raymond Damidian comes in our favor, is that the MRI was originally built on the premise that cancer cells and healthy cells had different voltages, and then that changes the water structure. So then if you want to look at the voltage of the cell, you can just do MRI. And people have shown within the past couple of years, actually, that the T2 times, just this parameter that you can measure on MRI, are correlated with the voltage of cells. So, and Gilbert Ling, with his protein ideas, because proteins create the mechanical properties of cells, also has linked voltage to basically every other property that you can measure biophysically, right? If I want to look at the tissue stiffness, this is also likely a correlate of voltage if Gilbert Ling's theory holds true, right? So then some ultrasound array could measure voltage proxies as well.
Sorry to go back into some granular detail here, but you mentioned water structure quite a few times, and I've tried to understand it. What exactly do you mean by water structure, and why is it important in the Gilbert Ling view?
Yeah. So there's a big debate around this within like the water physics community. And basically the idea is that if you look at a protein backbone in Gilbert Ling's model, there are the kind of, it's almost like a checkerboard of positive and negative charges. So you have this like, that is like partially positive, and then you go down the peptide chain and you have a partially negative, atom, and then partially positive, partial negative. So there's kind of like a partially positive and negative chain all across like a peptide backbone. So like on a protein, it's like a long string of positive and negative charges. And then a water molecule is a dipole. So what that means is that it also has like a positive and negative side.
Yeah.
So then if you take this like chain and elongated it in space, and then you put water molecules on it, one would face up, the other one would face down, the other one would face up, all across the chain, kind of like a checkerboard. And the distance between the partial positive and partial negative charges on the backbone of the protein are about the length of a water molecule apart. They're about the size of a water molecule. So then Gilbert Ling sees this as, a protein basically grabs onto, one layer at least of, this is like the conventional model is the hydration layer of these water molecules, and it polarizes them based on the charges. And then this layer of water molecules in Gilbert Ling's model will polarize the next layer, which polarizes the next layer, which polarizes the next layer, and goes on. And then he's done this work where he calculated the properties of this water, and he showed that like in the most perfect circumstance, where you take I think it's a silver chloride crystal, which shares the positive and negative checkerboard pattern. On a silver chloride crystal, you should be able to take like basically infinite layers of water, and it should polarize all of them. And this water should not freeze at any attainable temperature and should not boil until like thousands of degrees. And there's like some experimental evidence to show this, like in the most ideal possible circumstance. So he's saying basically that you polarize these water molecules, it changes the physical properties. And then the key thing in Gilbert Ling's model is that it excludes solutes. So if there's sodium that's trying to get into the cell, the water excludes it, because when the sodium tries to come in, it has to bind to the water molecules around it because it's a positive charge, and tries to orient the dipoles towards it. And it can't do that because the water is now oriented to the protein.
Okay, that makes a lot more sense. Thank you. I think I'm glad I asked that question, because understanding structured water is like the key to understanding. And thank you for that explanation.
Yeah, and I want to say a few things about this too, because there's like a lot of like structured water products out there, and I kind of think they're scams. I mean, like one of Gilbert Ling's
What is
things.
the structured water product? Like just a bottle of water? That's
people sell that. I mean, you can probably go to Aion and get like a $40 structured water bottle.
What does that even mean? I thought this was something that happens in. Okay, go on.
Well, yeah, like it should happen in cells, basically. I mean, the core thing, Gilbert Ling's model would not work if the structure didn't disappear instantaneously whenever you like change the protein structure, right? So people claim, because of some researchers like Gerald Pollack, that there's this like hexagonal water, like there's this weird structure of water that occurs whenever you put it on like a hydrophilic surface at all. And then this extends out pretty far, and then you can like harvest the water and put it in like a water bottle, and make it like grow plants faster, things like that. And to me, that seems like totally impossible. But I mean, I don't know. I could be wrong, but it seems impossible to me. So I would warn against like thinking of this as like a thing that you can eat. It's more like a mechanism of life rather than like a thing that can exist outside of like the vicinity of a protein.
Yeah, that's kind of what, yeah.
Yeah. And also like the average distance between two molecules in a cell, like two proteins, for example, is about 3 water molecules. So the ideal circumstance, you could have a lot more structured water, but like practically in the cell, it's probably like 2 or 3 layers of water that need to be structured. Like not very much at all. for this to be a real effect.
That makes a lot more sense. You need to have a bit of a vivid imagination about how a cell exists to be able to imagine all of this. But, if I can get there, I know other people can as well. Zooming out a little bit, one of the things you say in your white paper for Aion is that small molecule approaches have kind of reached their limit. So you have companies like New Limit trying to do epigenetic reprogramming, a bunch of others trying to, push the limits of longevity studies, but you think that's a dead end. Why specifically do you think that's a dead end?
Yeah. Well, so I think there's like multiple things here. So one is like small molecules, and I think that like companies like New Limit and others are doing more like genetic approaches, which are like slightly more promising. I guess I'll start with small molecules. So we've done this like analysis on, there's this database called the Drug Age database, where you can look at the lifespan results of basically all of the small molecules that have been tested in high quality lifespan studies on mice or other animals. So you can look at, does this extend lifespan in mice by 10%, 20%, 0%, whatever. And we've plotted all of them that have been replicated at least once, so like the higher quality ones. And you look at the curve that you get resulting from this, and you can like kind of fit a distribution to like the data here. And then what it tells us is that, the average lifespan extension that you get when you give a drug to a mouse and you expect it to extend life is around like 5%. And then the standard deviation is, I believe it's less than 10%. The number that we got to is that you would need to test like at least 30,000 small molecules to get radical lifespan extension, like anywhere close to 50% lifespan extension. And the amount of like money and time that it takes to like run a mouse study of like high enough quality, like $30,000 to $100,000 and 4 years. So if you multiply that by 33,000 at like the lowest end, I mean, some of the numbers that we got like with different fits got to like millions of molecules that you'd have to test. You're just like never going to get there by testing molecules in animals without some like massive change in approach. So I mean, like, yeah, there could be some arguments that like AI will make this better, but I'm like slightly skeptical. And then we can go to like, so it's like New Limit, and then these companies, I guess like Altos, the companies that are trying to do reprogramming.
Yeah.
So we totally buy into the idea that if you reprogram a cell, you make a cell more like a stem cell, it basically becomes younger. And this is like because, the reason why I buy into it is because people have done this in mice before. They've taken mice, genetically modified them to have these protein factors that reprogram cells. And then they activate them for 2 days, let them rest for 5 days, then activate them for 2 days and let rest for 5 days, and so on. And then they lived like 30% longer. So we already know that this can extend life in mice to a pretty significant degree, like something that's really hard to get with drugs alone. But the main problem with it is that you don't have like spatiotemporal control. So if you look at the voltage of all of the cells, right? I have like a bone cell that's a very differentiated cell, maybe like a negative 100 millivolts, very charged cell. And I have like a liver cell, which is negative 40 millivolts or 50 millivolts. And then I have a stem cell, which might be negative 20 millivolts. Well, I have to basically take the voltage all the way down from negative 100 or negative 40 and go make it be like a stem cell again. So if you view it like, you're doing this development pathway in reverse, some development pathways are longer than others. So to make my liver be reprogrammed takes like 2 days, but to make my bones be reprogrammed or my neurons be reprogrammed may take like months. So then if I decide to do this, if I decide to give myself these protein factors that reprogram my body, the only way that I can do it is by pulsing it in the way that they showed before, where you only reprogram the liver for a little bit, and then you stop before the liver turns into a big, like, undifferentiated bundle of stem cells that just kills you. And then you get no progress on the brain or bones or other organ systems. So then, yeah, and like what you die of if you just like leave the Yamanaka factors on, if you leave the reprogramming cocktail on, is you just die of multi-organ failure long before you get any like reprogramming in your brain, for example. So then there are possible clever genetic ways to solve this. One way that you could do it is, you could take Yamanaka factors or some reprogramming cocktail that like maybe is pretty efficient, and then you could make it sense what type of cell it's in. You could be like, oh, I have a microRNA switch that turns me on only when I'm in a liver cell and turns me off when I'm partially reprogrammed. But then the problem with that is that you need to create a different gene circuit for every different organ system and cell type in the body that all reprograms at different rates, and then deliver all of that perfectly. And then over time, ideally maybe you get rid of it after you dose this once, because cells will transdifferentiate, and then now your gene circuit is going to be like kind of in the wrong places, and you might start generating teratomas or like cancers, and once a single cell starts to differentiate, and then you reprogram it again in the future. So there's like all these challenges. You have to basically have like perfect delivery, perfect temporal control over a gene circuit, which is like already difficult. You have to make sure that your gene circuit can be like added and removed whenever you want, when we already have like immunogenicity problems of like repeat dosing with viruses. It just seems like there are too many challenges in front of it like working, because you're micromanaging like every cell in the body. And this is, what Levin has talked about is like the best way to go about making changes in biology is like apply the higher level tools of like psychology and neuroscience, or sociology. Would be like, how do I make these cells feel better, right? Instead of having to micromanage every underlying mechanism, just do like change a global variable and then prompt it gently and see what that does. We're both like not trying to micromanage everything. We only want, say, centimeter level control over the organ systems, because we think most organs are big enough aside from the brain to like, you don't have to be like perfect in that I have like cell level precision, but I have like organ level spatial precision. And we don't want to have to like manage this like chemical thing. We want to have like the perfect temporal control. So we can say, I'm going to turn on your liver reprogramming, and I'm going to turn it off when your liver's done, and I'm going to turn on your bone reprogramming and leave it on for as long as I want. And the natural solution to that is using fields.
It's so interesting to hear you talk. I think my original question was around why genetic reprogramming and small molecule approaches are a dead end. And I think you gave a pretty good answer, which is that you just simply do not have the kind of spatiotemporal control you need to truly enhance longevity in the way that you want. The better approach is to take this high-level, complex adaptive system view and manipulate these systems as opposed to individual cells, micromanaging these individual cells, which is the phrase you used. That makes a lot of sense to me. And I guess the challenge for you is to actually have that spatiotemporal control using fields and build the right devices and all of these things that are incredibly difficult, but it seems like the right approach.
Right. Yeah. I mean, it's like on an engineering side, it's like the most practical thing, right? There's like, I think like longevity has a few of these things where like scientifically there's something that's like extremely valid and like makes total sense, but then on the engineering side it's basically impossible.
Yeah.
And then there's things on the science side that there are like fundamental gaps, but are maybe possible, and on the engineering side is like trivial. So yeah. I mean, I think that once we have
where are you right now on that matrix?
Yeah, we've chosen that the engineering problem should be the trivial one. And then there's like basically one scientific challenge in front of us so far,
which
is like going forward. It's very easy to make a cell have a voltage that's closer to zero, but it's very hard to make a cell be more charged, essentially have a more negative voltage remotely. So I can just like shake up a cell, stress it out. The default thing is go closer to zero. But then how do I make the cell be more negative? And that's like the one challenge to solve remotely. And then after that, I mean, the visions of the machines that I've told you, I mean, it's basically like the same format as an MRI, just like control magnetic field gradients in space with some other signal, and then, or like have some robotic arm with an ultrasound transducer on it, or like some jello bed. Maybe you have to be in a jello bed. That's
I'm down. Yeah, fix my liver. Hopefully not anytime soon.
And even on like the field side, like there are things that are scientifically basically solved, but engineeringly, like on the engineering side, just not tractable. Like one example is light, like in the terahertz spectrum. So basically like light is, hundreds of terahertz in frequency. And then far infrared is like lower terahertz. And then, radio waves, like, so Wi-Fi is like low gigahertz, radio waves are megahertz. So like between radio waves and light, there's like the terahertz radiation. People have shown that they can resonate with specific ion channels. So they can like open and close specific potassium or sodium channels to control the voltage of cells. Like people have done this already. But the problem is the body is very, very good at absorbing this light. And scattering it as well. So then the challenge is like, how do I make this light penetrate any deeper than like the couple of millimeters that it does right now, when we want this to work in like like very deep organ systems.
Wow.
So like that's one example of still on the field side. It's like the engineering problem is like a super hard physics problem where you basically have to, you send light in, you measure all the light coming out, and you cancel out all the scattering with like destructive interference. It's like possible, but it's like a super sci-fi physics thing And then, but the size problem is solved because people have already been able to control the voltage of cells. So,
Right. I see what you mean. I guess I know you're hiring engineers right now. What would you say to an engineer who has experience in this field, in magnetic fields? How would you, I guess, pitch them on Aion and tell them to join?
yeah. So I think so. My grandfather, he was at Bell Labs for a period, and he like told me recently about like what it was like to be at Bell Labs, and he said that basically the thing that made it different from everywhere else was that there was like a ridiculous amount of patience where people were basically given their own projects. They were able to explore things that may or may not go anywhere for like multiple years. And then people invented things like radio astronomy just from trying to make like a better, radio transmitter and like trying to get rid of the background. And oh, what's that background? It's the cosmic microwave background of the universe. Oh, okay. But he said that yeah, there was like an immense amount of like patience. Everyone was given agency where they could like do basically their life's work, and everyone had a great sense of humor. And that was like what the three things that made Bell Labs Bell Labs.
Yeah.
And I think we have time. We're trying to move as fast as possible. We like to adopt that model where these are the things that there are many approaches towards this goal, right? And if you are an engineer and you have a better idea of how than I do of how to get to the specific goal, then we would definitely be willing to house you and have you try to get to this goal, right? Like the shape of the solution is definitely reading the body states and modulating the biophysics of the body in real time. But then how to get there, there are multiple ways. And we have our core paths, but we would be willing to add paths. Or if you're just mission aligned in longevity and love building things and don't really care exactly that it has to be your path, we can also I can bring you onto my theories and have you do the things that I want you to do.
That sounds awesome. I think it would be a real pleasure for anyone to work with you. It's always amazing talking to you. There's a few other questions I had. You're based in St. Louis. The company's based in St. Louis. It's not like the most obvious place for a biotech company to be these days, or for young biotech founders to be. Why St. Louis, and how are you liking it?
Yeah, I mean, I think St. Louis is pretty great. There is like a significant amount of just like biotech infrastructure here for I was surprised by it. There's quite a lot of lab space where the space that we're paying for is like literally less than a fifth, I think, of what people are paying for in the same amount of space in San Francisco, and like the access to talent and has been like fairly decent. Where there's WashU, which is like kind of Ivy League adjacent university here, where people would be willing to stay, but there's just like not so many opportunities here that people have seen. So we've hired people out of WashU already, and like I think that generally on a societal level, I think that like I'm slightly bearish on cities.
Yeah.
So I mean, I raised my initial round in upstate New York in a town of like 10,000 people, right? I was like 7.5 hours away from New York City, in a town right by the Canadian border called Potsdam. And yeah, it was like a tiny, tiny little town, but I raised money from Silicon Valley investors just online, right? So I think that like the enhancement of communication online has made the importance of being like physically somewhere like slightly lower. Like it's still great to get screen time, or not screen time, FaceTime, not screen time with people. But like I think the gap is closing over time, and especially maybe with like hopefully Mark Zuckerberg will solve this for us, right? Get us in VR in the same room. No, I don't know.
Yeah, man. Hopefully, like the crappy animations of people's floating heads. Yeah.
But but yeah, like I think that there's like a general type of person who wants to like not be in San Francisco,
Yeah.
where like you don't want to be in a place where you have to like just deal with the negative parts of cities where like I don't know, like insane amount of traffic and like homeless people, or I mean like the walkability is pretty great, but I walk everywhere in St. Louis, and the extreme like distraction of social lives that you may get to go to every party where you have to virtue signal in San Francisco. Like maybe you just want to have a quiet life outside of that. And I think that more and more companies are going to be built out of cities for people who want this. I mean, I think Elon has built his companies like outside of Austin or like in remote parts of Texas for SpaceX, right? So
Well, okay. To be fair, that was more of a geographical constraint. Like you can't build a rocket.
Yeah, can't build a rocket ship there. Yeah.
Yeah,
but I mean, Tesla and like the Boring Company and these things, they could be anywhere, right? But they're kind of like a little bit far out of Austin, right? They're not like in the city.
Yeah, no, that's right. There's like you need space, etc. But I think more importantly, to your point, it's the cultural aspect of if you're in SF, you're kind of drinking the Kool-Aid, and you're always going to be thinking like, oh, should I be working on that hot AI startup instead? Like,
Mhm.
what am I doing bioelectricity? And I think the kind of people that might self-select to go to St. Louis to join you are going to be a very special type of person. So I'm really excited for the kind of talent you're going to attract.
Right. Yeah. And I think over time, like the like barrier in front of travel, like also becomes like more marginal. Like, I mean, initially, like being scrappy, like you shouldn't be taking plane tickets every week. But then if you have like a Series C company, like your plane ticket isn't going to be so expensive that you can't get FaceTime with people that you want to or like need to for your to raise more money. So I think it's like more of a barrier at the start than like in the long term,
Yeah.
if that makes sense.
Yeah, absolutely. To wrap things up, I have a couple questions, or maybe three questions. What are some things that people simply do not understand about bioelectricity and the cellular nature of our bodies that you would want everyone to actually understand?
Did you say the top three things?
Well, just top three or number one, whatever it is.
Yeah. So I have the things that I think are the most interesting, and I think that the things that are most interesting may not be the best practical answer to this question, like what people should know, but these are the things that I would like to tell people. If that's,
Please.
I think Sydney Fox would be a great person to read. I think this is like the biggest like travesty in science that I've seen is like the lack of recognition of Sydney Fox's work. This guy Sidneyfox. I have a site called literature.bio. If you go
Oh yeah.
there, you could like
Yes.
download a couple of his books. Basically, like I think this is like the most convincing origin of life science that I've ever seen, where this guy basically took amino acids and he heated them up and then cooled them down, and then they formed these things called proteinoids, which are kind of like proteins but have like some slightly like branched off structure in like the wrong places. And then these condense into microspheres, which like look like cells. They're like these little like circular spheres that are all the same size. If you like do electron microscopy on them, they look like they have a membrane. And then he took these proteinoid microspheres and he's made them do everything that life can do. He made them synthesize DNA, synthesize peptides, like do chemotaxis, like go around and like find food. He found that they could have action potentials, like they were electrically excitable. So then, like thinking about Michael Levin, well, I think you mentioned to me before this, like something about the definition of a brain. So I think Michael Levin has this like idea that kind of mind is a like more continuum than it is like a discrete, just like step function thing,
Yeah.
where everything has like this, everything that's animate in some way, and maybe even like down to particles, but at least like starting at life, everything has a little bit of agency where like a cell will go around and try and maximize like the amount of food that it can have in this like small place. It has like a small range of cognition. And then you move up to like a dog, and a dog can understand a few hundred yards but maybe not the distant future, and then a human can understand the distant future. So everyone has like these different like kind of cognitive light cones, and it's like a spectrum of mind that goes all the way kind of like down to these like very simple things. So like it's Michael Levin would think that it's like not really disputable that cells have some degree of this like cognition. But then I think what Sidney Fox did is he showed that there was pre-cellular cognition in things that like looked basically like life, right? He was able to take these proteinoid microspheres, have them be electrically excitable, and transmit like propagating essentially action potentials between chains of these, like almost like a brain, but before cells even existed. So nobody knows about this guy for some reason, and I think that's like a travesty. So I think that is what I would recommend people looking at, just for like their own pleasure, and tell people this in parties that the RNA world ideas of life's origin are wrong.
That I need to read Sydney Fox. That you're absolutely right. Why is the RNA origin actually wrong? It seems compatible. I don't know.
Well, so yeah, I think the RNA world idea is basically they say that there's these like kind of like nucleic acids that can form these RNAs, and then RNAs can do a lot of the things that life can do, like catalysis. They can phase separate, so they can make things that kind of look like cells. But the problem that is in front of that is, for one, like how do they like kind of self-replicate, and then two, they're like very unstable in these phase separated things. But Sidney Fox like made these things that like actually did self-replicate
Mm.
and we're stable and we're all the same size. I mean, it's like possible that the RNA world idea is correct and maybe these things like co-evolved in some way. But if we're just like indexing on like what looks the most like a cell, the RNA world hypothesis is generating basically nothing that looks like a cell, whereas this guy has basically made cells like from nothing.
Got it. Got it. Um, it's funny you brought up the Michael Levin view of consciousness being a continuum. The more I listen to him, the more I realize his view really aligns with the Advaita Vedanta view of philosophy that I subscribe to. I don't know if you're familiar with it. It's non-dualistic philosophy. Basically, the idea it's a big branch of Hindu philosophy. The idea that consciousness is everywhere, and that you are not separate from the ultimate consciousness, which is God, if you want to call it. I think that view necessitates a continuum of consciousness between different living entities. And it makes a lot of sense to me on an intuitive level. Last question for you. Did you have a thought?
Oh yeah, yeah. Bernardo Kastrup is probably who I would say is like the metaphysical version of Michael Levin in this respect.
What's his name?
Bernardo Kastrup,
Bernard.
Bernardokasrup. He has like a couple PhDs. He worked as a he worked at CERN before. I think he had a PhD in like computer science, and then now he got a PhD in philosophy, and now is a an idealist. He thinks that basically, I think the most succinct summary is that there are no quanta, there are only qualia. Is the most succinct summary of his ideas. So like the position of a particle is stored in qualia, and like everything is qualia.
I don't even know if I want to go down that rabbit hole right now. I'm gonna read up on him and then come back to you with some questions. That sounds super interesting. You've already, provided all of these recommendations of things that we should read. And is there anything else that you would recommend to anyone listening that they should definitely read that has been instrumental in shaping your view?
I think I think Ray Peat's articles are great, just like from a practical perspective in life. I mean, the way that I found Gilbert Ling was through Ray Peat originally.
Yeah.
I was researching in my PhD like the kind of things around like water physics and how water interacts with proteins, but I didn't hear about Gilbert Ling until I read Ray Peat, and I saw this guy Gilbert Ling in discussions with all the other early 1900s German colloid chemists who also talked about water physics back then. And then that's like what really put me on the trajectory that I am now. So I think that like Ray Peat's articles are great just from like one learning about nutrition in a slightly heterodox way, but also maybe there's some other like scientific connections that could help jumpstart other people's like, I guess, thought journeys.
Yeah, I had a small detour down the Ray Peat rabbit hole and started drinking more carbonated water, sparkling water, because apparently that's
Nice.
better for you. But there we go, there we go. Yeah, I need to more fully understand his view. One thing I didn't ask earlier, and this is like completely not related to any of this, how is the cosmetics business going? Because I bought one of your products.
Mm-hmm.
You have a great brand called skin.food that I would recommend to everyone. How is that business going, and why are you doing that?
Yeah. It's going well. I mean, I basically just wanted this product to exist for myself. Like my skin was pretty bad. It's like not great right now, but you should have seen it before this product.
Yeah.
I mean, yeah, my skin, I've always had like terrible acne and just like bad skin, and every skincare product has just made stuff worse. So I like wanted something that I knew was going to be good, and I just formulated it. And I felt like the difference between me making it for myself and then making it for, making it a company was just buying the, like multiple gallon quantity of the oils rather than just like the few ounces that I needed.
Yeah.
So I did that, and then I got some branding done, and then made it a company, and now it's like growing decently. I'm not like putting a ton of time into it because like Aion is the main thing, of course, but it exists and it's growing. So whenever I get some inspiration at night, I'll like do some work on it and try and get some stuff out. And we're going to be releasing like more stuff pretty soon. We also released an oral vitamin E product, and hopefully we'll be releasing some more things.
That's super cool. How did you decide that that was what you needed? Or you had the formulation had vitamin E, DHEA, and one other thing.
Yeah. Yeah, and squalane. So basically, I found that whenever I took from Ideal Labs, like Georgy Dinkov, like his company, I took their vitamin E and I put that on like a pimple or my acne, it would immediately go away like the next day. But it was just like so unpleasant to use because vitamin E is like so thick and sticky. It's just like not nice to have. So I just wanted a thin version of vitamin E that wasn't bad. And like squalane is also pretty like inert to my skin. So so I just combined the two to make it like the highest possible purity vitamin E that I could get, because most of it is full of seed oils still, if it's thin. So I took like the very thick, high purity vitamin E that has no seed oils in it, and then I combined that with like an inert oil squalane. And then also at the same time, I found that like all these studies that showed that DHEA was like super anti-aging to skin, but I also wanted to supplement like a little bit of DHEA every day. You know, it was really hard for me to get it from like an oral version because like the doses that everybody like would sell in these pills was like way too much or had like weird like cut things in it. So I just wanted to like make sure that, you know, the source of everything was good and these were just like the needs that I had at the time, and it turned out to be like a very nice product.
I love using it. I think it's helping me as well. So thank you.
Awesome.
This was awesome. Is there anything else you want to leave the conversation on, or do you think we kind of covered everything?
Yeah, I think we wrapped it up pretty well. Again, yeah, I think Michael Levin just won on Lex Friedman yesterday, so I would encourage people listen to the new podcast that he just did. It was pretty good.
A hefty podcast, like 2.5 hours.
Yeah.
Classic Lex Friedman. Yeah, but I'm excited to listen to it, and I'm excited to keep up with everything that you're doing. It's just so fascinating, and people can find you at anabology.com, right?
Yeah, that works. Anabology.com, anabology on Twitter, or Aion.bio. So that's AION, which is a like Greek god. That's why it's not ION. It's not for AI, even though maybe when AI takes off, we can pivot to be AION.
Man, oh man, yeah. Once you have that, you will have like some ML involved, I'm sure.
Yeah. Probably more like reinforcement learning is what we're thinking at this point, but it'll be something cool.
100%. Dude, awesome. This was great. Thanks so much for taking the time. I really appreciate it, man.
Great. Thanks.
All right, dude. Chat later.